Okay, so now we're going to walk through the steps here of what happens. And I'm going to talk a little bit about the calcium, the role of calcium here, but we'll talk a lot about calcium when we get to excitation, contraction, coupling. So we had this actin protein. We described this protein already. We described the regulatory parts of this protein, tropomyosin being that thread-like protein in purple, and then troponin being the pink complex that had 3 binding sites. Now, before we talk about this process, I want you to understand that this doesn't happen if there is no calcium. So in the absence of calcium, this process does not initiate. When calcium comes into the sarcoplasm, into the cytoplasm of that muscle cell, it can flood the actin and it can bind onto its binding site on troponin. Okay, and when it binds there, then it stimulates the troponin to drag tropomyosin, that thread-like protein, away from the binding sites, and that uncovers the binding sites and makes them available for myosin to then reach up and bind. So understand that if there is no calcium, these binding sites are covered and actin cannot interact with myosin. This needs calcium in order to be initiated. Okay, sorry. So let's say we had our end plate potential on muscle that generates a flood of calcium into the muscle cell. Calcium is now binding onto its respective binding site on troponin. That uncovers the binding sites here, that uncovers these myosin binding sites. And then myosin now is free to reach up and grab onto this protein. So the first part of this is myosin starts off in its high energy form. Right, we talked about the 2 forms of this myosin head, and when it has that phosphate and that ADP bound to it, it's really attracted to actin. It's going to reach up, it's going to bind to that binding site, and then it is going to release that phosphate off, okay? That phosphate is released off, and then we have the power stroke, okay? So the energy is then donated to this process, and myosin drags actin towards the M line, okay? Myosin binds to actin, and it essentially moves this way, dragging actin with it. After that, the ADP is then released off. Once the ADP and the phosphate are released off, myosin now goes back into its low energy form. But there's sort of a caveat here. Myosin gets into its low energy form, but here it's in what's called rigor. And rigor simply means that it gets a little bit lazy. It's stuck in this position. It's not going to let go of actin until a new ATP is available to bind. And this is why we have rigor mortis. So after death, the tissues, the muscles stay exactly as they are. So if someone died holding a cup or hugging or whatever position your muscles were in, they will stay that way indefinitely because of this rigor. Because there's no ATP, right after you've expired, your body's no longer making ATP, and so all of your muscles stay in this contracted state. In the presence of ATP, it comes in, it binds to this myosin head, and now it can be released. It's back into its low energy form. It doesn't like actin, right? That attraction is now broken. It's not wanting to reach up because of the presence of ATP and the fact that it's now in a low energy form. Right? And then this process will continue again and again. That new ATP will now be hydrolyzed. And as soon as that's hydrolyzed, it's again in its high energy form. It's going to reach up in preparation to bind to actin again. Okay? So this cycle of how we make contact with actin, drag it towards the M line, release contact, make contact again, drag it, release contact. This is called cross-bridge cycling, okay? So myosin is the cross-bridge, it makes contact with actin, and this is how we facilitate this process.